DNA is usually shown as a double helix, but it does not always exist in only one structural form. In a eukaryotic cell, a very long DNA molecule is packed inside a very small nucleus. So DNA has to coil, bend, and adjust its structure depending on the condition around it. There are several forms of DNA, such as A, B, C, D, and Z forms, but the three major and commonly discussed forms are aDNA, bDNA, and zDNA. We will start with bDNA because it is the most common form found in living cells under normal conditions. bDNA is the usual Watson-Crick DNA structure. It forms in a highly hydrated environment, which means when enough water is available around the DNA. bDNA is a right-handed double helix with a wide and deep major groove, while its minor groove is narrow and relatively shallow. It has about 10 to 10.5 base pairs per turn. But what's a turn? When the DNA helix completes one full rotation around its central axis, we call that one turn. This means the helix twists around once and reaches the same facing direction again. But one turn does not mean one base pair. This is one base pair, but one turn requires many base pairs. In the case of bDNA, it's 10 to 10.5 base pairs. The rise per base pair is about 0.34 nanometers, and the pitch is about 3.4 nanometers. Pitch is also known as the vertical length of one complete 360-degree helical turn. The diameter of bDNA is about 2 nanometers. In bDNA, the base pairs are almost perpendicular to the helix axis, with only a small base tilt of around 6 degrees. aDNA is also a right-handed double helix, but it is shorter and wider than bDNA. The major groove of aDNA is narrow and deep, while the minor groove is broad and shallow. You can imagine aDNA like bDNA being compressed from the top. When it becomes compressed, it becomes shorter in length and wider in diameter. aDNA usually forms when DNA becomes dehydrated or when water availability is low. It may also form in conditions with a higher concentration of ions. aDNA has about 11 base pairs per turn. The rise per base pair is about 0.29 nanometers, and the pitch is about 3.2 nanometers. The diameter of aDNA is about 2.6 nanometers, which makes it wider than bDNA. Its base pairs are more tilted compared to bDNA, with a base tilt of around 20 degrees. So, compared to bDNA, aDNA is wider, shorter, and more compact. zDNA is very different from both aDNA and bDNA. Unlike aDNA and bDNA, zDNA is a left-handed double helix. The major groove of zDNA is almost flat, while the minor groove is narrow and deep. It is called zDNA because its sugar phosphate backbone follows a zigzag pattern. zDNA usually forms in specific DNA sequences, especially sequences with alternating purines and pyrimidines, such as GC-GC repeats. It can also form when certain proteins bind to DNA and force it to adopt a different shape. zDNA is longer and thinner than both aDNA and bDNA. It has about 12 base pairs per turn. The rise per base pair is about 0.37 nanometers, and the pitch is about 4.5 nanometers. The diameter of zDNA is about 1.8 nanometers, making it the thinnest among the three forms. Its base tilt is around 7 degrees. So, in simple terms, bDNA is the normal and most common form of DNA. aDNA looks like a compressed, wider form of bDNA, and zDNA looks like a stretched, thinner, left-handed form with a zigzag backbone.